Good afternoon, everyone. Sorry from our side for making this overlap with the Seeing is Believing conference. That was, well, can't think about everything, but this talk is for sure the more exciting one, so you should stay here. So Adrian, I asked him about a short CV, and I will just read it because it's really short. So he's finishing his PhD in Anna's lab. He's already looking so mature, hard to believe that it's only doing a PhD. So he likes working with deep learning-based segmentation methods, is interested in generalization, transfer learning, and learning with sparse supervision, something that we probably all like. And in his spare time, he runs, weight lifts, meditates, and raves. So he's a raver. That's cool, I guess. So maybe you can demo it at the end a bit.
I wrote it 10 minutes before the start, yeah.
Okay, Adrian, go for it.
Okay, thanks, Christian. Thanks for the introduction. Thanks for showing up. I see many familiar faces and some unfamiliar, so that's really, really nice to see new faces. So my name is Adrian, and like Christian said, I'm finishing my PhD, and I'm from Anna's Kreshuk lab at EMBL, and I'm going to talk about my recent research, which is also available as a preprint on Archive. We have a new version, so that preprint is kind of outdated now, but we are going to upload a new version pretty soon. So I assume that people here will have a very different background and interests, so I'll try to give a short context and motivation for this work before diving into the method itself. And maybe one more thing before we start. Ulrike mentioned that already, but maybe you didn't hear it. Please feel free to interrupt me at any time, because I know that these Zoom talks are pretty one-sided in nature and asynchronous. So if you have any questions, just unmute yourself and ask questions if anything is unclear. So the title, Embedding-based Instance Segmentation Method with Limited Supervision. So let's dive in. When I started my PhD, I was working on a segmentation of 3D light sheet and confocal images of plant tissue. And here are examples of those two imaging modalities. The confocal image is a 2D slice of confocal image of Arabidopsis ovule on top, and light sheet image of a growing lateral root formation, also from Arabidopsis in the bottom. So those are large 3D image stacks, which we try to segment into individual cells. And we use the pipeline that is well known for people from connectomics who are working with images as well. And such pipeline consists of two steps. First, we train a neural network that predicts the boundary probability maps from those noisy images. So it's a simple semantic segmentation task, right? And in the second step, we take the boundary image, we over-segment it with something like a distance transform watershed. In essence, we split the image in many small regions that we call superpixels, and then we build a region adjacency graph from those superpixels, in which every superpixel is in a node in the graph, and the edges connect neighboring pixels, right? And finally, we solve a graph partitioning problem, which is NP-hard in general, but for that bit, we can use greedy strategies like greedy agglomerative clustering, or we can put additional consistency constraints on the partitioning and solve a multi-cut problem, which solves it until optimality. It also has an exponential complexity, but very good approximate solver exists. So the two-step pipeline, first step differentiable, second step is a graph partitioning non-differentiable step. And although we would love to have something which is completely end-to-end learnable, this proves to be extremely difficult to develop in neural networks, which are permutation invariant and whatnot. And there are pros and cons of such a pipeline. The advantage is that given that the network was trained correctly and gives a high quality prediction mask of those boundaries, we can obtain an excellent segmentation results with this pipeline. It's also very fast. So predicting boundary maps with neural networks on GPUs is an embarrassingly parallel problem. So once we have the network prediction, which can be run in parallel, we can also parallelize the watershed and graph partitioning. So that's also a nice addition. But there is a crucial disadvantage, which is the dense annotation bottleneck, right? So in order to train a powerful neural network, we require a large amount of densely annotated data, usually annotated by the experts, because especially in bioimage, and this is costly. So that's just to give you an example. A single light sheet. This is just a 2D slice of the 3D image, but a single light sheet stack of growing root primordia that was acquired in Alexei Maitsev lab in Center for Organismal Studies in Uni Heidelberg. It took a student around 4 months of work to segment this stack. We bootstrapped this segmentation from a more shallow learning methods. We used Elastic, but also like this proofreading and all the time that it required setting up the tools. It took 4 months to do such a segmentation. Another example is those beautiful obvious from Kay Schneid's lab from TU Munich. They have 48 stacks like this. Here it's nicely shown like the 3D image of the input confocal. In this left part, here is the boundary prediction by the network, and here is the final segmentation. This was a bit easier problem than the previous images, because the images are not as noisy, but it also took a significant amount of time to proofread those stacks to provide this 48 volumetric stack, which we use later for training. So yeah, this is very very costly. And maybe again, just to recap the standard segmentation pipeline. So again, 2-step, right? It's important that the first step, we have a differentiable function represented by neural network. And the second step is non-differentiable, which is the graph partitioning, which gives us the final segmentation. And I was asking myself 2 questions, like, can we bring this whole pipeline closer to end-to-end? So how can we overcome this non-differentiability? And can we also use other representation apart from just the boundary map that will allow us to overcome this annotation bottleneck? How can we train from sparse labels? So how can we train it with just a few cells? In this case, annotated. And I tried to tackle both of these questions, but mostly focus on the second one, which is the representation of the image that could be learned from sparse annotation. When we talk about meaningful representation of something, we usually have in mind some vector or representation or some embedding of that thing. So what I experimented with is pixel embeddings or embedding networks, which arise from various losses formulation. I used contrastive loss, which I'm going to show later. But in this setting, just briefly, a neural network maps each and every pixel in an input image to high-dimensional vector space, right? So each pixel is going to correspond to a high-dimensional vector. And in such a way that pixels that belong to the same instance, they will be close together in this vector space. And when they belong to 2 separate instances, that will be far apart in this feature space or vector space. So this is just for visualization. The original vector space given by the network was 16-dimensional. We PCA projected it to 3 principal components and treated it as an RGB image just for the visualization. So this is like compressed embedding space. when we have such an embedding space for a given input image, we can also use standard clustering algorithms like MinShift or something density-based like dbScan or even k-means if we know the number of clusters that we're looking for, and just segment it. This is also obviously non-differentiable, right? Okay, now to train the pixel embeddings, how do we actually do that? So here is the image from the seminal work from De Brandbadere et al. And the discriminative loss or contrastive loss that was introduced there. It consists of two terms, two forces at play. We have a pull force, which puts together pixel or embeddings of that pixel that belongs to the same instance, right? So in instances, let's say that instances are color coded. So the pull force will pull everything to the center of cluster in the embedding space, okay, for pixels that belong to the same instance. And then we have to counterbalance this pull force so that we don't have a collapsing, like everything collapsing to a single point in the feature space. So we have the second force, which is a push force, and we push apart the center of clusters. We don't push apart pairwise pixels over every instance because that would be too expensive. We push apart the center of clusters. And also it's important to know that both of these forces are hinged. So basically they only active up to a certain distance. So if the pixels from an instance are put together close enough to the center, we don't penalize this anymore. Okay. Similarly, if the cluster centers are far apart enough, right? If they're far enough in the embedding space, we also don't penalize it. You see this hinges. This is the pull hinge by the smaller circle, and the push hinge is the larger circle. This is important for us because, especially this pull hinge, because I can use it to and freely sample one pixel or one embedding, take this hinge around it, and extract the instance. So this is a trick that I'm using extensively in this work. And the push hinge is important because you know it gives like a lot of freedom to the network, and the embedding space doesn't explode too much when we start pushing around everything. Below you can see how those clusters evolve in the embedding space. So here we projected for visualization into 2D space. We start from embedding space, which are quite random. So the pixels are behaving quite randomly in the embedding space, but then as we train with those with these two forces, they start to form clusters in the embedding space where each cluster corresponds to a given instance in the image. Here is the form of those two terms defined. The pull force we penalize when a given embedding for a pixel is too far from a cluster center or some anchor. We also hinge it, and there is some normalization by the number of pixels, number of instances, and the push force. Similarly, if two cluster centers are too close together, we also penalize that. And now I've extended this basic loss formulation by additional losses, which enable me to train it from sparse labels. And I want to train it such that I don't lose too much quality of the embedding space that would be given by the fully supervised setting.
Adrian, I think we have a question by Sebastian. Sebastian, do you want to unmute yourself and maybe ask a question in person?
Yeah, I haven't seen that. Sorry.
Yes, hi, Adrian. I was just wondering, how do you assign the instance to each of these points?
Yeah, so the instance, so it's coming from the ground truth, right? So this is still a fully supervised setting that I'm talking about. So I'm reviewing like how would you train embedding in fully supervised setting, right? So you do it, so you would take pixel, for example, let's take this instance. You would take all those pixels from this instance, right? And you would just pull it to the center of the clustering embedding space. So let's say that's mean, right? So basically, the network would predict some embedding for every pixel here, right? And you would like to cluster it together. That's how we take it. Okay,
perfect.
and then
Thank you.
for two different cells, you would push them apart. Okay.
Thanks.
Yeah, no worries.
Yeah.
Maybe I could ask something too, since we're kind of stopped. Is that okay?
Yeah, no worries.
Here in this case, I guess broadly the question that I'm wondering about is this loss, I think, is such that every instance, every cell in your case will want to go to a unique point in the embedding space. Is that true? And I guess, could you relax that? Mostly I'm thinking maybe you don't care that far away cells go to the same point in embedding space, since you'll be able to spatially disambiguate them. Is that a reasonable idea? Maybe you don't need this. Yeah, maybe this is overly unnecessary, but I guess I'm just wondering what you think about that.
No, you're totally right. I mean, you can imagine it also depends on the post-processing. So yes, normally if you train with that, each cell or each instance will have a unique space in the will correspond to a unique place in the embedding space. Yes, that's true. You can force it or normalize the embeddings to lie on the sphere, right? That's one way you can do. Or if you want to be even smarter, you can actually use the spatial coordinates, right? And maybe something like a 4-color theorem. It says that you can paint with any map with just 4 colors if you're in 2D, okay? And then you would need like just a simplex with three colors, right? In the end. So yes, but that would require some post-processing. But yes, it's a good question. There might be, you might get away with some instances falling to the same region in a way, but this loss prevents it. So I'm not sure if that answers your question. So would you? So intrinsically, this loss actually prevents 2 instances falling into the same region, right? But you could think of a formulation that can use like 4-color theorem to still be able to segment.
Oh, that's helpful. Thanks.
Okay. Okay, I was here. So before we move on of how to extend this basic loss, this discriminative loss, let's talk about a differentiable instance selection. Okay, this arises from the formulation of the proof force. So I'm taking in this case a sample image from the CVPPP data set, which is a very common data set for leaf segmentation, right? So people want to segment each individual leaves in this data set. And let's say that given the embeddings from a network that we train, we want to extract a binary mask for a given leaf. Let's say like this leaf here. Okay. So what we do, we randomly sample an anchor point inside this instance. Okay. We take the embedding of this point, right? Because we know that now we can find like the embedding vector for this pixel. And what we do, we compute a distance map from this embedding to all the embeddings in the image, right? So basically everything inside this instance hopefully should be very, very close to this anchor, and everything else should be far apart, right, in this distance map. And then as a final step, I use a kernel function to soft threshold the distance map and get a probability map. So you can think of it as a probability that a given pixel belongs to that instance. Okay, and this so this can be given an anchor. This can be done in a Fourier differentiable way. just a formula, and this is the RBF kernel that I'm using for on top of this distance map. Those are embeddings for each pixel. This is my anchor embedding, right? And then the sigma, importantly, we can compute the sigma based on this pull hinge that I've shown you, right? This circle, this hinge on the pull force called delta V. And then there is like one important hyperparameter in my algorithm called T. I call it the kernel threshold. And we require a kernel value which is delta V apart from the anchor to be at least T. Okay, so maybe we show you, if I show you the diagram of the kernel. If this is my anchor at point 0, right? This is my delta, or this is my hinge, right? Here, we can basically choose different T's and get a different, basically threshold with delta V apart. So what it does, basically, with higher T values, we get a very high sigma. So what it does, it's making the mask very, very sharp. Okay, that has some benefits, but also if it's too high, this T value, right, we're starting to peak on pixels which are far away from the instance of interest, and this is becoming unstable. I'm going to show you basically briefly what does it mean. So this kernel threshold T parameter, if we take some fluorescence images of cells, okay, we take one cell, we have the embeddings, we take an anchor, and then for different threshold parameter using this kernel, this is what this soft mask would look like, right? It's quite blurry. If we increase the kernel threshold, it's going to be more binary. With 0.9, it's almost binary. But then if we increase it even more, we start to pick up, we assign non-zero probability to pixels which are outside of this instance, and this is very bad for training and makes this whole thing unstable. We got best results with T equals 0.9, and then there's like a lot of ablation in the paper. This is important hyperparameter, actually. Yeah. So this is about the kernel. Okay, so let's finally get to this sparse object annotation. So what does it mean to be sparse? So, and how can we extend this basic contrastive loss to work in sparse setting? So now let's take that our training data looks like this. Given the input image of a bunch of leaves, we have labels only to some of them, right? Some of leaves are labeled. This is something that human would do, right? I would, if I would try to label this, I would pick leaves one by one instead of, going row or columns and trying to assign labels, right? So now we have some of the leaves annotated, and then there is this huge unlabeled region in black, which contains the background, the genuine background, but also the instances that that weren't labeled by the annotator. But what we know about this unlabeled region is that it should be far away from the objects for which we have the labels. So what I did is I introduced another push term when I push all those pixels from the unlabeled region. I push them apart from the clusters for which I have the labels, right? We push them apart in the embedding space, and this turns to be quite important addition. This is also hinge for preventing overfitting and whatnot. And another extension of this loss of the space loss is what I call the instance-based loss, and here is where this differentiable instance extraction comes into. So again, input image, we get the embedding of this image. And now we have some labels for some instances, right? We sample and anchor points for each instance, because we know the ground truth, right? So we can sample randomly some anchor points. And now what we do is we extract, for each anchor, we extract the soft mask with our differentiable instance extraction procedure. We stack them together and we compare it to the ground truth with a loss of choice. So we in our work we chose a dice loss, but any sort of like overlap loss would work, binary cross entropy and stuff like that. Dice loss has nice properties of being very robust to pixel imbalance, right? We also experimented with other instance-based losses, not only BCE or dice. We can replace this function, this loss, with a discriminator, which is learned with adversarial setting, and try to find something like a shape prior for an object. This proved to be very beneficial for very stereotypical objects like cells in plants, which are very similar, but it doesn't work across the board. But still, it's a nice set of experiments. I won't go deeply into that, into the adversarial setting, but it's described in the paper. So we have those two additions to the loss, but there is a problem. And the problem is with the complex background patterns. So since now we have only, some of the objects labeled, here's only some of the leads and none of the background, right? So the network is completely unconstrained in this unlabeled region, and it starts to pick complex patterns in the background. So now let's say that we use only 10% of ground truth leaves for training. And although the network was able to correctly embed pixels on individual leaves, it doesn't consolidate the background anymore, okay, to a single cluster. And this is quite natural since we haven't constrained the network here. We just told the network to push this away from the instances that we have labeled, but the convolution network wasn't constrained in this region, and it naturally propagates the patterns that were found in the background. Here, this pot that should be in the background, it's clearly visible. And then in the inference step, when we try to cluster, this is a problem because we recover this false positive spurious instances in the background. And this is, again, because the network was not constrained in the unlabeled region, right? We just pushed apart this region in the embedding space. In other modality, when we use EM images, which have complex cellular ultrastructure in the background, here we're interested in segmenting those mitochondria. But again, because of this complex background patterns, now the problem is that after clustering, we actually lose some instances, right? So we have false negatives. So it's even worse. So how to overcome this? And I think this is the most important addition, an idea that I have in this work, and something that I call embedding consistency loss. So again, we have an input image. Now we do have two neural networks, okay, which we train with those sparse annotations only. And what we do is first we augment this image such that it preserves the geometry. So we don't rotate, we don't do any elastic deformations, but what we can do, we can do blurring, adding some Gaussian on Poisson noise, doing some color jitters on this image. And we pass this augmented image to our embedding network, the first one, and this is going to result in some feature space, right? We do the same again by augmenting different random augmentation which preserve the geometry. We pass it through a second neural network, second embedding network, and now we don't have any supervision in an unlabeled region, right? But what we can say is, hey, I want the representation of this image in the feature space given by those two networks to be consistent, right? So if I take one anchor somewhere, and I take the neighborhood around this anchor, and I try to cluster it with this mass extraction procedure, it should give me the same instance, right? It should make those embeddings consistent. We should make those embedding space consistent. And this is the idea. So again, we use this differentiable instance selection algorithm, but now we select anchors only in the unlabeled region. We extract the mask for every anchor, and we apply a consistency between those, right? So each and every anchor will correspond to some instance, right? So this embedding space will give us some instances for those anchors. This embedding space will give us different instances, and we want them to be consistent. So maybe this is not a very convincing picture because those instances look very similar, but you have to trust me that they are a bit different, right? Again, we stack those different instances together and apply a dice loss, right? So those are the masks given by the first network F, we call them. Those are instances given by the network G, and we apply the dice loss on top of them, enforcing this consistency. And so what is the second network? So although we could use two separate embedding networks, initialize, differentiate, and train simultaneously, I chose the momentum contrast scheme. So the second network is implemented as a momentum-based moving average of the first network. Okay, so if theta G is the parameter of the second network, and theta F is the first network, this is like the slowly moving average. This is the update rule of this G network. So by setting this momentum parameter to be very large, the second network is slowly trying to catch up with the first network. This is something that's been explored in self-supervised classification methods. It hasn't been tried in a dense setting, and it has this beneficial computational efficiency because we can back probe only to the first network, and this is just a moving average. And this is not something random, right? The second network, this wasn't my first choice. This momentum contrast network, and here I show only qualitatively different variants of those of this network that we can pick as a second network for the consistency, using those two simple test images for the CVPPP. Again, this is something that you already seen. If we don't have consistency loss, again, network starts to pick up on complex background patterns. We want to avoid it. At first, my first try was to use a shared network. So basically, the second network is just the same as the first one, and we just rely on this augmentation to slightly perturb the embedding space and improve the consistency. This somehow consolidated the background, but you still see that it's not a one consistent cluster, right? The background is not one cluster. Another idea was to use a dropout. Basically, the first network was again a copy. The second network was again a copy of the first network, but we randomly dropped out some neurons in the bottleneck of the unit that was producing the embeddings. And this was somehow better, but still produced some artifacts in the background. So it still didn't perform that well. Then the third variant was the trained variant, where the second network was completely trained from scratch, the same as embedding, the first embedding network. So that was the most computationally intensive. It somehow worked better than the shard variant, but it wasn't that much better. actually, and the momentum contrast, actually, you can see that it improved a lot, the embedding space. So basically, it was able to consolidate this background correctly, right, and get rid of those spurious patterns in the background. So that was something that we still trying to understand. There is like some theoretical work of why this works, why it doesn't. We explore it more experimentally. Do you have any questions to this part? Maybe?
I have a question.
Yeah.
It seems like that you invested a lot of work in this. Wouldn't it be easier to get, like in an Elastic that I sometimes use, to get a feedback on the failure mode and then somehow add ground truth in the places that matter, like this complex background?
Yeah.
Would more ground truth help here? More than...
Yes, it would. I mean, this problem is structured as this positive unlabeled setting. So I never have any labels in the background, but it would definitely help. Like I didn't actually explore that, but you could try to have the labels like scribbles in the background, okay, and treat it as a separate cluster and try to push it away somehow. And that might work. Yes, I agree that this is something that might be explored. But this is a very specific setup where you only have this the objects labeled, and none of the background. Okay.
Got it. Thanks.
Yeah, but I agree. This is if you have like some scribbles, then that might help. Yeah.
So maybe just a very brief other question. So right now you quite nicely show how the different steps are applied to, in particular, these planned images. How diverse was the set of images which you actually used? Like, was this also more also on the molecular scale, like closer to all these type of images, which or imaging which goes on in cell biology and neuroscience, or so which type of data sets have you used in order to do the training and to come up with your algorithm?
Yeah, so I'm going to show you that show that at the end when I'm going to show some results. But I tried different modalities. Yes, that was there was confocal, there was light sheet, electron microscopy images, and also those natural images. And now we're also working on some like fully natural images like cityscapes and whatnot.
Yeah, perfect. So I was just too early with my question. Thank you.
Yeah. So it's we try to make it quite generic. Maybe just to finish this talk. So this thought about the consistency part, right? And seeing the ablation. So if we train the network with just 10% of ground truth objects, 10% of the leaves, you can see that with consistency, you nicely get rid of all those spurious patterns, and then the segmentation is much better. The same goes for the EM images, where here we train the mito-EM on the mito-EM data set with just 5% of mitos, and we were able to get rid of those spurious patterns and get much better segmentation results as well. So this is the complete full picture of the training procedure. We have those two embedding networks now trained with the modified contrastive loss. We have the anchor sampling procedure. We label separately. We sample anchors from the labeled instances, the blue dots, and we also label the unlabeled region, the yellow dots. And we introduced those two important losses. One is the instance-based, which works on the labeled objects, and then the consistency loss, which works on the unlabeled region. And together we train this with, like I said, with sparsely labeled object without losing the quality of the embedding space that would be given by the fully supervised method. Maybe just let me briefly show you some loss ablation results on this CVPPP data set. there are two metrics: the symmetric-based dice, which higher is better, and the difference in countics on the leaves. Fuse tried different sampling, anchor sampling strategies. They all performed roughly similar, so this is not that important. But the bottom part of the table is more interesting. So here we trained in the sparse setting with just 10, 40, and 80% of the objects. And we try to remove either this push term or the consistency term, okay, and show what happens there. And in particular, we can see that in the sparser setting, when we remove the consistency loss, we get a much more detrimental effect on the final segmentation than when removing this push force, this additional push force. When we get more and more labels, this consistency term becomes less important, which is more natural, like closer to intuition. Maybe a few words about the inference. So what happens when we have the embedding network and we can produce an embedding for a given image. So we're still exploring this part. We can use a classic min-shift clustering with a bandwidth which is set to this hinge parameter on the pull force. We can use a density-based method like HDB scan. We also introduced in the paper something that is called consistency clustering. I'm not going to go too deeply into this, but briefly we start with some segmentation method which has a high recall. So basically, which can segment, which gives you all the true positive instances that are there, and then you slowly filter out a noisy cluster or what we call unstable cluster with some clever procedure. This is described into the paper, and now we also explore the graph-based partitioning scheme, where we construct the affinity graph with edge weights that are based on the embeddings given from the network. And we do that because those three methods are quite slow for large images. Okay, so and you know, graph partitioning, we have people in the group constantly work a lot with scaling up this method. So it's just nice to have something quicker. Some qualitative comparison between those three clustering methods. If we take again CVPP data set, we have three test images on the left. This is the PCA projected embedding. And here you can see that min shift, it's nice because it recovers all the instances, but unfortunately it also has a lot of noisy clusters around the leaves. HDB scan performs the best. Consistency clustering performs roughly as good as HDB scan. And this is unfortunately not the case for something like data science ball, where we have a lot of cells with different sizes. Here, HDB scan completely underperforms. You know, you will have to. There is like one hyperparameter that you have to tune, and you would have to tune it to the size of the cell. So this is a bit you know, hyperparameter dependent, so to say. Min shift has again very high recall. It recovers all the cells, but you also have this noisy instances around it. And in this case, this consistency clustering was performing the best. So there is still some room to improve for improvement in the inference, in the post-processing, actually. Some quantitative results on the 3D data set. So here is the 3D Arabidopsis ovirus. This is just one patch of it. The embedding network, you know, the embeddings are really, really sharp, and the segmentation is almost perfect. What's important, however, I think the most important thing here is that we've just, by the way, the acronym for our method is FOCO, right? So that's how we called it. And importantly here, when we computed the A-round error of the final segmentation, we're training just with 10% of the cells labeled. We get a better results that this fully supervised embedding method from the Brown-Badera. So this is already quite nice result. And we get state-of-the-art. We combine SPOCA with an adversarial setting. So this is the obvious. We also tried our method in transfer learning setting when we try to fine-tune this OVU network, the embedding network trained on OVU data set, and try to apply to a different data set of Arabidopsis stem cells. Okay, and so those images look very different from the obvious, but it looks actually really good because we've just 1% of labels from this stem cells data set, right? With 1% of objects, so to say 1% of cells. We get ARAN, which is almost 2 times better than the obvious network only. Okay, and as we add more and more supervision, it gets closer to the fully supervised network trained on a STEM data set. This is the 3D. We also tried this transfer learning setting on the EM data sets. Here the MITO EM, a big, very big EM data set. We train it in 2D actually. This is a 3D data set, but we train it slice by slice just by simplicity. And we showed that we get a positive transfer with our methods. So what we did is we bootstrapped the network, the embedding network. We trained with a very small EM data set of mitochondria, also called VNC. And you can see that this VNC network, embedding network, if applied to this new data set, it performs quite poorly. So this is the ground truth, this is the embedding space, and this is the final segmentation. But if we fine-tune it with just 1% of mitos from the MITO EM, we get really great average precedence scores already. In fact, if we would train with just 1% of cells of the mitos directly on EM, the scores would be much lower because it would overfit a lot. Here are some quantitative results. Again, you've seen this image. This is if we remove the embedding consistency loss, so we get all those complex patterns in the background, and the segmentation again is underperforming. Interestingly enough, for MITO EM, just 5% of labels used for training our methods gives segmentation scores which are on par with like fully supervised settings. So this kind of opens also quite interesting questions about how much labels is enough for training a good segmentation, a good embedding network, right? This is something that we also would like to explore in the future. This is one avenue of active learning. So basically, someone asked about like how to pick an instance that would give you the most gain in the segmentation. Yeah, so that's actually quite interesting question, and we would like to pursue this. I think this adversarial learning setting, when we have a GAN setting, would be quite good for that. So basically, active learning, showing the instances. We have a human in the loop, how to pick instances that would give the most gain for the segmentation. We would also like to explore the possibility of completely self-supervised pre-training. So basically, use this embedding consistency loss just for pre-training, self-supervised, without almost any ground truth, and use that to as a backbone for a dense segmentation task later, be it instance segmentation or semantic segmentation. We'd like to try it out if this works, actually, if this idea works there. And last but not least, we would still try to improve the clustering. You've seen that different clustering methods perform differently on different data sets, and we would like to try to unify that and find the best method that works across the board. So this is the roadmap for future work, and with that, I would like to thank those beautiful people that helped me with my work, Chin, who ran a lot of experiments constantly, obviously, and Anna. Yeah, so it was a great ride, and thanks for joining and choosing this talk instead of Seeing is Believing. And I'm happy to answer any questions if you have them.
A quick question. How high dimensional was the embedding space, and is higher dimensional always better?
Yeah, that's a good question. Yeah, I usually I picked embedding space to be 16-dimensional in all the experiments that you've seen. It's quite interesting because higher is not always better, but what I explored is when I have the embedding space, I explored what are the actually how much variance is explained by the first principal components, and what we can see, what I've seen in across data set is around 5 or 6 principal direction, right? In embedding space explain like 99% of the variance. So you could probably reduce it to something like 8 or 6 and get very good results. But anything lower than that would be probably detrimental. But also higher is not better. Yes.
I have a question.
Yeah.
Sometimes microscopy images contain more than one channel. Have you guys considered using more information about structures in order to maybe the question is, how easy was it to combine more information about the structures that you have in order to do this segmentation issue?
Yeah, so
let's say you have, for cells, you have a nuclear stain and a cytosolic stain or something like that.
Yes, that's a very good question. So if you have a nuclear and a membrane stain in 2 separate channels, that wouldn't be, there is no like very straightforward way to incorporate this information. Actually, this is something that Chin started to work on, right? This is because we were so encouraged by this work. We're now trying to actually have something like a self-supervised setting that would allow us to incorporate the nuclei and the membrane information together without almost any supervision, and try to set up the learning problem in a way that exploit this consistency, that you have just one nuclei enclosed by the membrane. So in this setting, no, you cannot easily do that, but potentially there are ways to set up a learning process, like I said, to exploit this multi-channel information. Yes.
So can I?
It's a very good question and a very good research direction. So, okay.
Do you hear me? Hello.
Yeah. Hi, Constantine.
Okay, I just wanted to quickly remark on this, because I mean what you said, right? It's all correct, but I think there's it depends a bit on what you're interested in, right? So what you said is basically if you're interested in both the cell and the nucleus segmentation, then you want to learn sort of from each other during this. And this is, I think, very interesting. And like you said, Chen is thinking about this. The sort of simpler setting is that you just have extra channels, but you still just want the cell segmentation. And I think then the easiest way would just to concatenate the channels as input to the network, or maybe do a sort of a bit more later fusion, right? And that might already help you quite a bit, because it just is more information that the network could use.
Yeah, that's a very good point. Yeah, we don't have multi-channel data in our experiments, so we didn't try that. But yes, that's a good point. Yeah, potentially the embedding network could pick up on information from different channel and improve. Yes.
Yeah, it's about the network structure, right? The fact that if you just concatenate it, might not understand that there's a pixel correspondence to the same place in the image. It might pick it up somehow, but it might be good to tell it that these things are actually multidimensional and represent the same place in space somehow.
Absolutely, yes. Yeah.
Maybe I've also still two questions. So one question, or the first question would be, so you motivated everything by mentioning that the segmentation of a specific data set took previously a couple of days. So how fast is right now? A couple of months even, yeah. So so I guess still for the training part, one needs a couple of hours, but how fast are you on average with the data sets which you were presenting so far? So that's the first question. And then the second question would be, just from the point of usability, I mean, I guess right now people who listen to the talk and you will think, oh my God, it's pretty amazing with 5% of the data to actually get already such nice results. How can I get this run on my own data set? If you could just say a little bit about how people have to navigate through the space in order to manage their own data sets with your presented tools, that would be great. If you could just say a few words to that as well.
Yeah, definitely. Thanks, Ulrike. Yeah, that's a very good question. I mean, so the time estimate, it's hard to say, you know, still hard to say how much annotation is enough for a given problem. So, for example, for leave, we found out that okay, 10% is actually enough to get to the point of the fully supervised previous embedding method, or even surpass that. For mitose, it was 5%. So it's very much data set dependent, but I mean, you can always iterate on this, right? So you start with some instance label, try to train a segment and see where you get, and then you can, you know, manually pick the instances that were segmented and repeat the cycle, so to say. We did in terms of usability, yeah, we try to use it at EMBL at very challenging mouse embryos, actually, in the data set, the light sheet data set of mouse embryos. And that was actually quite challenging for this method. So we had a student that tried to label. The data was very noisy, and she did like really very good job trying to label nuclei and cells, but the embedding space wasn't you know, quite the decision boundaries weren't that clear, right? So it would be good to develop some sort of method which says where, like, get an indicator of how much data is enough actually for a given data set. That's why this avenue of active learning and yeah, and you know, trying to inform the user of which instances to pick would be good. In terms of just using it, you know, the using is simple, right? If you want to use the method as it is, you just take your data set, label some cells, and just throw it and try to hope for the best, right? I think this important question is like, how much is enough? This is something that we don't have an answer to yet.
Maybe just as a user suggestion, if you know elastic people, combining this with an elastic GUI, right, would be, yeah, I think, the most helpful for users. I don't know if there's a, you know, one of the nice things about some of the workflows in Elastic is the ability to have a uncertainty map.
Yes. Yeah.
Right, that could come from a lot of steps in your pipeline. It seems like it has a bunch of ways where it checks for self-consistency and can tell you where it gets confused. So yeah, I think that would also really help for getting as many people to try this.
Yes,
I would certainly be happy to.
Mm-hmm. Yeah,
and maybe I can, sorry, just quickly on that point, right? So, I mean, the whole group is working on this model zoo effort, just sort of making pre-trained models more available, but also overall getting some infrastructure to run this in more user-friendly tools like Elastic or also Napari. And I mean, this is, I think, very much on our goal to integrate this method and also other things that are integrated in the group, either in Elastic or Napari. And hopefully this is not so far in the future, but I think right now, you would be stuck with setting this up yourself and just running the PyTorch code. But at some point, hopefully this will work much easier out of the box.
Yeah,
exactly.
We very much appreciate it as users, that effort.
Awesome. Yeah, there will be work also, like I mentioned, you try to explore if this would be possible with very, very little supervision or no supervision at all, right? With the right sort of data augmentation and things like that. And then if you have such a model pre-trained, right, then you can load it in Elastic, then label something and have like this quick feedback of how your embedding space changes, how your segmentation changes. You can validate it visually. That would be very helpful. So basically, we have something that runs somewhere, in self-supervised way, right? Like it's training itself, and then you load it into this trained model, like Konstantin mentioned. It's somewhere in the model zoo. You load it, you label just a few cells, and then you improve on that. Yeah. Because the representation that you've learned with the self-supervised method is already powerful enough. Yeah, that would be the ideal world. We're not quite there yet.
Yeah, I think with that, Christian, I think we are at the end, at least of the seminar part and of the Q&A session. But so let's thank Adrian one more time, maybe, for his presentation. So for me, the only thing which is left to say is maybe to give you an outlook of which type of talks will happen, or which type of upcoming talks are ahead. So we actually do have already in 2 weeks a very interesting talk ahead by Niklas Zofreniov. I'm pretty sure I've butchered the last name. He's from CCI, and he will actually give a presentation about Napari as well. That was also a talk initiated by Christian Tischer. So thank you very much for that as well, for highlighting this. And then the talk after that, so meaning the talk in a month, will be actually given by Carson Stringer. And I'm pretty sure she will talk about cell pose and also other ways to segment and analyze data sets. So we swapped this talk with Karl Svoboda's talk because I think he had other things happening at the same time. So his talk will be postponed to January, but Carson was so nice to fill the gap, and we're actually very much looking forward to these 2 sessions. And so again, thank you very much, everyone. And but for those who would like to stick around, because we still have the Meet the Speaker session, which is right after this talk right now. So if you would like to talk with Adrian, or if you would like to get to know a bit more about the software, if you'd like to learn how to use this for your own data sets, for all in-depth questions, you can stick in this channel for a bit longer. Christian and I will also be here, so you're not alone with the speaker. And yeah, maybe also just to get to know Adrian or ask a little bit more questions about also the lab he's working in, or even EMBL. So feel free to stick around a little bit longer. Otherwise, have a nice day, and see you on October 19th when Niklas will give his talk about Napari. Okay, bye-bye.